Hello, I'm Scott Coates. I'm the Senior Director for the AOAC Research Institute, and today I'd like to talk about the Research Institute and the Performance Tested Methods program. So, AOAC Research Institute is a division of the AOAC International. It's a nonprofit international scientific organization, and we provide an independent third-party review of test kit performance. So the companies that are approved are licensed to use the certification mark in advertisement and packaging in 1-year increments. And we review the test kit annually to make sure that it's performing the same way it did when we approved it. And we're making sure that if there are any changes that are made to the test kit, that we know about it and that we've approved it, reviewed it, and evaluated it. And the test kit companies get to use this performance tested certification mark. We're very international. The origins of our test kit methods are from the United States and outside of the United States in pretty much equal amounts. We also have a wide range of independent testing laboratories. We have mostly in the U.S., but there are a lot of outside of the U.S. Testing laboratories. Same thing with our expert reviewers. We have a pretty good balance between the United States and outside of the United States. And the type of test kits that we review also pretty broad. About 70%, maybe closer now to 80%, are microbiology. That we have toxin, antibiotic residues, and biochemistry. We have approximately 5 phases of review. The first phase is consulting, which takes about a month. The second phase is when we get an application in from a company. A company can do consulting, but not obligated to submit an application to the PTM program, or probably all of them do. Phase 3 is data collection. That can take a wide amount of time. If all of the data collection goes right, we can probably do it in a month and a half. And when all of the data is collected and put into a report, that's when we send it to the expert reviewers. The expert reviewer may take a month or more. Sometimes we get into a process that takes longer because some of the data did not support claims and we need to work that out. We can the test kit company can either add additional data that shows us that it does work, or we might require that a particular performance claim in a particular matrix, the test kit doesn't actually work. And then the 5th phase is publication in our journal of the AOAC and in our inside management, laboratory management magazine. Phase 1 consulting. It costs $2,000 for non-members, it's $1,000 for members, and a $500 rebate when the company submits their application to us. The Phase 1 consulting is all optional. The test kit company could go directly to Phase 2, but most companies choose to use this consulting because we do a lot of work. We do it all the time, so it's very efficient for the Research Institute than it is for a company to put together a package. So with the application, the test kit company sponsors submits an application. It's a form that we have on the website. They submit a package insert or user's manual, and they may call it one or other name, but the instructions on how to use the test kit. They submit a QA/QC synopsis and then also application fees. At that point, when we get the application, we will work with the test kit company to determine the expert reviewers. We have a pool of expert reviewers that we can use, and we can let the test kit company know who's an expert reviewer, and we can work with them to get expert reviewers. We also work with the test kit company to assign independent laboratories. We have a list of recommended laboratories on our website, and we can share with the test kit company the independent laboratories. Phase 3 is data collection. The test kit company can do in-house data collection following protocols that we've developed during a consulting project. And there's an open line of communication between the research institute and the sponsor. So at the same time, we can be doing independent testing supervised by the AOAC Research Institute. We're carrying out a testing protocol that we developed during a consulting program with the test kit company. Those protocols are pretty detailed, and I'm not going to go through all of the requirements, but I'll go through the most important ones, which are specificity. If it's a quantitative method, that gives you a specific result, such as 1.2 gram per mil, then we need to do specificity, limit of quantitation, comparison to a reference method, matrix, and accuracy. If it's a qualitative method, that's giving you a plus or a minus. We usually check for inclusivity and exclusivity, repeatability, percent method agreement with a reference method, recovery, and matrix. So you can see there's a lot of technical requirements, and also to make it more complicated, some of the methods are microbiology, so they need to be taken into consideration with the microbiology. Phase 4 is when we get all the data back together, both the in-house data and the independent testing laboratory data. We merge that into a consolidated validation report. A review package consists of that consolidated validation study report, package inserts and user manuals, raw data from the company and the independent lab. What we do is we send that out to reviewers, and we give them 2 weeks to review, and then depending on what the expert reviewers tell us and the report that we get back, they decide whether to grant or decline approval. Phase 5 is the publication. So we certified as a performance tested methods. The kit name is immediately added to the list of performance tested methods maintained on the AOAC website, which I'll show you in a minute. Method validations are reported on the website, provided on the website, and a second validation report is published in the Journal of the AOAC. So this is what our PTM test kit looks, PTM approved method test kit database looks like. You can search for the analyte, you can search for the matrix, you can search for the test kit name, you can search for the company. So it's a very useful list for people that are looking for test kit for a particular purpose. We've had a lot of experience validating microbiology rapid methods. We've done more than 350 reviews on a wide variety of food composition, chemistry, residue, toxin, and microbiology methods. And we have more than 300 reviews of rapid microbiology test kit. And it currently lists about 350 microbiology test kits that have been granted performance tested methods. We also have a lot of experience with all the different target bacteria and also the ATP as well for hygiene. So we have validated test kit for Listeria, Salmonella, E. coli, coliforms, total plate count, Campylobacter, and ATP-based hygiene methods. We also have a lot of experience validating different methods, techniques. We validated cultural methods, immunoassays, polymerase chain reaction, enzyme method, bioluminescence, chemiluminescence, and electrical resistance. So it's a lot of different types of test kits that we have validated. And also a lot of international experience. About 50% of the companies are based outside the US. About 30% of the independent testing lab is performed in Europe. About 25 of the expert reviewers are based in Europe. We do have a cooperative agreement with NMKL, and we have a working relationship with Nordval, Microval, and AFNOR, the French standardization organization. We also have ties to ISO, Codex, and WHO. So looking at all of the validations that we've done, we do not always approve test kits, or sometimes the test kit companies need to improve their test kits before we can approve. So very common problems are incomplete instructions, either being too vague or leaving too much room for interpretation. And often important controlling parameters are not just prescribed. Also, methods are not fully adapted to new matrices. So for example, a clinical method often developed for serum or blood, applied to food matrices that are much different in protein and also other components. So that these test kits that are working for clinical purposes have to be adapted to food matrices, and it may be a different test kit. Also, many rapid methods rely on existing enrichment procedures to recover bacteria, and they assume that the accepted enrichment procedure will deliver the goods in all cases. And again, this gets back to the fact that a food matrix is different than a serum or blood matrix. A lot more competitive microflora that might inhibit the target bacteria, and some serovars do not reproduce at a fast enough rate, produce detectable levels of target bacteria. Other two problems that we find is that the market-driven desire to shorten incubation and develop time becomes a problem, because most strains and serovars are detected, but some of the strains and serovars cannot reproduce or metabolize fast enough to generate a signal. And then some test kits are just not ready for prime time. The companies have issues with manufacturing and quality controls. So if we get a couple of batches to review, it might vary from batch to batch because the manufacturing and quality control are not really good enough. But the good news is, in my experience from 30 years of experience, is the level of sophistication has materially increased. Some of the companies really do understand what's needed, whereas some of the companies just don't. And I see that as a combination of Darwinism and the market of evolution and the impact of regulatory and validation program. And the overwhelming majority of companies really want to do the right things. After all, their reputations are on the line. We have some new developments we're expanded the performance tested methods to include cannabis, meaning marijuana and hemp. So we did 27 different test kits last year to add marijuana as a matrix to their test kits. We also last year evaluated COVID-19 test kits as an emergency response validation program, and we validated 9 test kits for COVID-19 on surfaces. Also last year, we started the Reviewed and Recognized program. This is a very interesting program. It does everything that the Research Institute Performance Tested Method program does for test kits, but in this case, we're doing this for technology providers who have application notes or users notes. So typically, we find that technology providers are issuing these application notes, but there's no program to evaluate them. So that's what the Reviewed and Recognized program does. It's for application notes. And we have our first R-squared, we call it, approval this month in October. And we've got a number of application notes and technology providers going through the program. So you will be seeing this application note certification routinely a lot. So here you see the upper left-hand corner of the R-squared logo and the Reviewed and Recognized program. That's our logo. And like I said, this program is specifically designed for ensuring the performance claims and reliability of application notes and laboratory developed methods. And what we will be doing, very similar to with the performance tested method note, there'll be a certification that's updated annually for every certified method and provides a description of the method and underlying technology, a summary of the validation data, a list of specific food items and ingredients that were validated, the reviewers and independent testing laboratories that were used. These certificates will be on the website and they will be downloadable and can be included in the laboratory's accreditation document. On the right-hand side above my picture is what the certification will look like and what the certification logo looks like. These will all be, for the beginning, these will be in our test kit database. And if you type in a name such as ABC Company, then that will show up through the list of approved notes. So additional information resources, you can go to the website, AOAC website, and there is a list of performance tested methods with links to kit company's website. And you can see the link in the bottom there where the RI website is available. We also have application and data requirements and microbiology validation guidelines are available at the website. So the AOAC Research Institute team, we've got 7 technical consultants, but I'm the managing director, so that's the first, I'm the senior director, that's the first picture. Nora Marshall is the senior manager for the AOAC research program. Dr. Sharon Brunel is one of our technical consultants. Maria Nelson is another technical consultant. Linda Johnson is our technical consultant, primarily for chemistry type test kits. She's based in Los Angeles. We just recently added Dr. Cheryl Lassiter, and she'll also be sort of focusing on the chemistry part. We also have Dr. Dawn Dowell. She has a PhD in public health, and she does a lot of our annual review work. And then we have Dr. Don Gilliland. He's got a PhD in chemistry, and again, also worked with us on the chemistry part. And the reason why we added a couple of new chemistry technical consultants is because we are planning that our R-squared program will start to bring in many new methods for the R-squared certification, so that we'll now be in a position and have the appropriate expertise to evaluate these methods. And then general contact information is AOAC Research Institute at Gaithersburg, and you'll see our telephone number and our fax number.